Hello, I'm Nathaniel Williams, and I work in Carl Olson's lab at Carnegie Mellon, recording neural activity in visual cortex of awake viewing rhesus macaques. The poster I'll be talking about is titled Contribution of Receptive Field Center and Surround to Repetition Suppression in Macaque Visual Area V2. In primate inferotemporal cortex, a high-order area of the visual pathway, when a large and complex natural image is presented twice in succession, first as a prime and then as a probe, neurons exhibit repetition suppression, responding less strongly to the second presentation than to the first. This has often been assumed to arise at the level of inferotemporal cortex because these neurons possess the large receptive fields and sophisticated stimulus selectivity necessary for recognizing the image as a repetition. However, we recently discovered that neurons in the lower order visual area V2 exhibit repetition suppression under conditions identical to inferotemporal cortex. This raises the question, how do V2 neurons show repetition suppression to whole images when they have receptive fields which encompass only a fraction of the image? One possibility is that they are sensitive to repetition not only in the classical receptive field, but also in the surround. To assess this possibility, we monitored neuronal responses to sequential displays in which we controlled independently the repetition of elements in the classical receptive field and in the surround. For each neuron, we mapped the location of the receptive field using a grid of horizontal and vertical bars, and its size by taking the diameter of the disk, eliciting the strongest response as its extent. We also assessed surround responses using annuli, which varied in inner diameter. For the experiment itself, each stimulus consisted of a disk scaled to and centered on the receptive field and an adjoining annulus with an outer diameter of 8 degrees. The disks and annuli were taken from different natural scenes. Each trial consisted of a prime, a delay, and a probe, each 300 milliseconds in duration. The animal was required to maintain central fixation during the entire trial. There were 4 match conditions. Both the center and surround were a match. The center was the same, but the surround was different. The surround was the same, but the center was different, or neither the center nor the surround were the same. Taking the no-match condition as a basis for comparison, we measured suppression as the reduction in firing rate observed under each of the other conditions. We also presented match and no-match trials in which the disks and the annuli appeared alone, which I'm going to be showing first. Here's a peristimulus time histogram of the population mean firing rate over time of neurons recorded in V2 during trials when the center and surround were stimulated in isolation. The no-match conditions are the dashed lines, and the match conditions are the solid lines. The center alone condition is in red, and the surround alone condition is in green. When either the isolated center image is repeated or the isolated surround image is repeated, there is only a very modest hint of repetition suppression. The case was different, however, on trials when the disk and the annulus appeared together. Here's a peristimulus time histogram of the same V2 neurons, but on trials when both the center disk and the surround annulus were presented together. The no-match condition is the dashed line, the center match condition is in red, the surround match condition is in green, and the both match condition is in blue. The colored bars at the bottom of the histogram indicate times when each condition showed statistically significant suppression by a cluster-based permutation test. If we zoom in on just the probe response, we can see that, using the no-match condition as a baseline, in which we should see the least suppression, neurons exhibited robust suppression when both the center and surround matched the prime. There was also a strong effect when the center alone matched the prime, even when the image content in the surround was not a match. The surround match condition was indistinguishable from the no-match condition. However, there was a strong effect of repetition of the surround when the center was also repeated, as indicated by greater suppression in the both match condition compared to the center match condition. This is quantified by taking the difference between the center match condition and the both match condition, and is significant by the cluster-based permutation test as indicated by the magenta bar. We next analyzed the strength and timing of each effect by plotting the difference scores, as shown here. Shaded areas are one standard error above and below the mean. We measured the time at which repetition suppression under a given condition reached half its maximum, which is indicated by the colored triangles, which occurred at around 100 milliseconds for the center match and both match conditions, but markedly later at around 200 milliseconds for the additional surround suppression generated by the surround matching, above and beyond that from the center matching on both match trials. In conclusion, this work has shown that repetition suppression in V2 is sensitive to content outside the classical receptive field. The contribution of the surround is accentuated when content in the classical receptive field is also repeated. In addition, the late onset of the surround contribution suggests mediation by lateral connections in V2 or vertical feedback from downstream areas. Finally, I'd like to thank my lab, the members of the Kohn lab I share space with, the NIH for funding this research, and thank you for your attention. Please contact me via email at npwillia@andrew.cmu.edu or online if you'd like to talk more about my research. Thank you.